who and where are they? So the first one up is, where is this castle located? If you all want to just push the bar, if you know where I'm talking about, and yell it out, so we'll give you a...
Istanbul?
No, it's not Istanbul.
Okay.
All right, it's the Valletta, which is the capital of Malta, and the Victory Day is a national holiday at the city. And this Victory Day was to celebrate the historic sieges of Malta by the French in 1800, the German and Italians in World War II, and King George of the United Kingdom in 1993. It's a very scenic place. Where's this bridge located?
France.
All right. A little more specific. It's close by. It is on the, well, I can't, why? I'm going to the next slide, except I can't do it. Oh, it's on the bridge over the River Tera in northern Montenegro. It was completed in 1940 as World War II was spreading. And this was one of the arches there on the bridge was blown up by the Italians during the war, and it was rebuilt in 1946. It's a very scenic location. What is this mountain called, and where is it located? Anybody have any idea?
These must be the $2,000 clues.
Mauna Kea.
It's one of our longest allies of the United States ever.
France.
Exactly. It's called Bald Mountain for obvious reasons at Mount Ventoux, France, also known as the Beast of Provence. It was deforested, that's why it's brought, in the 12th century for shipbuilding purposes. So they cut down all the trees, obviously, and never grew back. How about the, let's see.
Hello.
The fjords and where's the location? Name what they're called, and where are they?
Norway.
The Cove of Spires in Alaska. They were created by the National Interest Lands Act in 1980. And what happened was when we declared that land pristine land in 1980, it doubled the size of the national park system in the US, doubled it for that. All right, how about this? Name the people in the village.
Benjamin Fisher and Richard Thompson.
You got it right there. These Dogon people are from the West African country of Mali. They inhabited it since the 3rd century BCE. If you don't know what that is, it's before Christ. This region was a UNESCO World Heritage Site and has been inhabited, as I said, there by the 3rd century, and it's known for intricately carved wooden masks. They're on the left, you can see those. Well, anyway, you have a little geographical knowledge. Now, are you in tune, Mike Newman, with the thought of the day?
I'm on the phone. I can't get into the computer.
Well, we hear you. Why don't you go ahead with that?
All right. Thank you. With Dr. David Stover presenting his craft talk today, I offer the following thoughts. A prayer for healing a loved one. Dear God, I sincerely pray for my loved one who is going through a tough time with his disease. Bring the power of healing and restoration into his body. Fill him with your restorative spirit so that he may experience healing and wellness. Lord God, bless his doctors and nurses, enlighten them and guide them as they do their job. Lend them your healing touch and use them as instruments to get my loved one through his illness. As David knows, persistence works. There's the old saying that as a river cuts through rock, not because of its power, but because of its perseverance. In our service above self, may we all be there in person for those afflicted with illness to lend a helping hand. I'm very pleased our club has David as a member. I wish him all the best and success.
Thank
Thank
you,
you.
Mike. Very good. Very good.
Yes.
Finally got you. Okay. Now we will have David Stover. Let me give the introduction. It's my pleasure to introduce you today, one of our newest members to the club, Dr. David Stover. David is a Ph.D., earning his doctorate in biochemistry at the University of Washington after attending Duke University, where he has earned his B.S. degree in zoology. David is a biotech entrepreneur with over 25 years of experience in drug discovery and development in oncology. He currently serves as president and CEO of Nammi Therapeutics, and also serves on the board of directors for several additional biotech companies. Most recently, he was the site head of the Angenesis, a Santa Monica-based Astellas affiliate, where he was responsible for 240 employees in the research, clinical manufacturing, and clinical development focused on antibody drug development. Trust me, this had nothing to do with the Wuhan labs in China. Prior to that, our esteemed Dr. Stover was the head of oncology biologics at Novartis. Earlier, David was director of biochemistry and a first employee at Kinetics Pharmaceuticals, a small molecule kinase inhibitor company that was acquired by Amgen in 2000. And then he was vice president of drug discovery, MCS Proteomics Company. In addition, his extensive experience in preclinical and early clinical development, David is an accomplished scientist and researcher, having co-authored 50 manuscripts in peer-reviewed journals. Now that I've probably mangled many of these technical terms associated with his obvious impressive scientific background, I want you to know he has yet to find a cure for the common cold. With that, here's David Stover for his craft talk. David, you're up.
Thank you, Tom. I think I'm done now. Good job for me. Can I share my my screen to put up slides?
Tom, you have to go into participants over to David's name and make him the host.
Do I make him co-host here?
Or the host, whatever is popping up. I think it's just going to say host.
Mark him as co-host so that you don't log yourself.
I stopped sharing as co-host.
So make him the host. He'll do his presentation. When he's done, he'll make you the host back.
How do I get him to be co-host?
So are you hovering over his name and you see where it says more and it says make a host?
I've got him. No, I don't want to mute him, but
Make him a host.
make host. Okay. You want to change the host? Yes.
Perfect.
Okay.
Okay, David, you should be host by now.
Hey, can you see slides?
There we go.
So I guess to start, I'm going to start with a caveat. So I really didn't know what was expected from a craft talk and never had an opportunity to see one done. So around and didn't get a lot of guidance from people, so I went online. And online, I found a number of examples. And it was interesting because all the examples I found, of course, have these very dramatic stories. So there's one of somebody that was picnicking at the Grand Canyon, and a boulder decided to start rolling and rolled over their leg, crushing their leg and pelvis and carrying them toward the edge of the canyon. And it was just about to roll over the canyon when it was stopped by a tree. And from that day on, the person had an affinity for trees and planted trees all over the place, really got into to tree planting. And that was their life story, was really about trees from then on. So I don't have any stories like that. So I'm going to share what I have, but it's no dramatic things like that on the way. A little bit of background bio on myself. I was born in Syracuse, New York, on the shores of Lake Ontario. My father, Raymond Stover, was a Ph.D., had a Ph.D. in particle physics, and worked at Xerox for his whole career. Interestingly, her maiden name was Taylor, so she was Elizabeth Taylor. Had a bachelor's in sociology and was a high school teacher. 3 kids. Older sister is Debbie and younger brother Michael. You'll see them in a moment. And I'm divorced with 1 daughter, Rania. She's 18 now. Those are the basics. My early years in Webster, it's a happy time for me. I lived there for my first 18 years. Great parents. This is the house I lived in. As you can see, it's very cold. Rochester was always the one of the top 3 cities in the snowfall. I was always looking to get away to sunnier places. Here's California. As a kid with my brother and older sister. So it was happy childhood, but nothing dramatic. But many aspects of that childhood have stayed with me over the years, like my love for animals. So started early with had a horse as a kid, dogs, cats, hamsters, and that stayed with me. I don't have a porpoise, but I kissed one, and have had cats and dogs ever since. Here's a ribbon I won as a horses. I sailed competitively with my father, Snipes. That's a 2-person sailboat shown here. You start a race with everybody lining up and trying to get as close to the start line. Of course, you have to go with the wind, so it's always difficult to get. In varsity football and basketball, but my competitive sports are limited. I like outdoor activities though, of all kinds, hiking and biking, and sports are more limited to corporate picnics these days. Cases, so obviously Halloween's, New Year, particular dance, wearing Galabea in Egypt, and a 60s party. And then the student. So I went to Duke University, as Tom mentioned. I went actually thinking I was interested in marine biology. And so they have a marine lab. So I went to the marine lab for a semester, and despite really loving it, I decided after that I really wanted to go into something more molecular. Ph.D. at the University of Washington. Graduating from there, went to work at Ciba-Geigy, which is now Novartis, a large pharmaceutical company based in Switzerland. So I went to Switzerland and for 4 years, here as a postdoctoral studies. And at that point, I met a woman from Egypt who I ended to the US and having my job. Things didn't work out in the end. After 25 years, we divorced last year. A very proud father of my daughter. She's now 18. This is just a few lower right corner. Cherish all my days with her over the years. Moving now more into my career, as I mentioned, I started off at Novartis, Ciba-Geigy, where I started as a research scientist, laboratory leader, then went on to director, and actually started the oncology biologics group for Novartis. So in 2003, Novartis decided to get into biologics, and so I was able to be at the forefront of that. And during my time at Novartis, I was involved in development of 2 drugs that actually made it all the way to Taz is an HIV protease inhibitor, and Gleevec is a kinase inhibitor for cancer. And I'll talk more about through each of the different companies I was in, the smaller ones, but I feel like I really made a contribution to the field and advancement. So at Agencis, which was a subsidiary of the Japanese pharmaceutical company, 10 years, mostly as the site head that was running the research, clinical development.
This guy's speaking, so he's not going.
8 new drug applications. So when a drug moves into clinical, tested in patients, you have to apply to the FDA for a new drug, and so we brought drugs to the clinic during my time. And one of those, Padcev, is a antibody drug conjugate, and I'll explain what that is later. That is now approved for metastatic bladder cancer. And then currently, I'm the helped found in 2018. We have 6 employees now, and we are developing novel therapies to treat cancer, really focused on using the body's own immune system to attack the tumors. So I'll go into a little bit of information, not too detailed, on each of the drugs that I was involved in that got approval, because I think each one of them tells a story that may be of interest. So this first one is Reataz, what has 2 names, a brand name and the generic name, Atazanavir. So it's an inhibitor of an enzyme that is required for the HIV virus that causes AIDS to replicate. So if you inhibit that enzyme, the HIV cannot replicate. So what it does is it processes the proteins that allow the virus to form properly and to bind the cell and get incorporated into the cell. Virus, and this is of interest, of a lot of interest these days, because this is basically what the cell, through that receptor that it binds to, it gets internalized, and the receptor that it binds to will determine which cells get affected. So COVID is binding to receptors on mostly epithelial cells, so you get it in your nose and your throat and your lungs where the epithelial cells are. HIV does it on T cells, so it's attacking the T cells and killing T cells that are necessary for your immune system. So when you have HIV, you're killing T cells that are system, and therefore you don't have an immune. Okay, so my responsibility within the program was actually So the idea was there were already two HIV protease inhibitors on the market, one from Merck and one from. What we were trying to do is because there were resistant mutants that were forming based on those protease inhibitors, we wanted to make one that would inhibit those resistant mutants. And so we were trying to be responsible for testing and coming up with the assays to make sure that it actually was doing what we wanted it to do. So now it is approved and it's part of the HIV cocktail that's made up of 3 different drugs that have really allowed people to HIV for many years and lead more healthy lives. And so now HIV, if you're able to get this type of cocktail, is no longer a death sentence. Interestingly, I thought Novartis actually dropped this program just as we trials because of the political and social controversies around HIV and AIDS at the time. This was back in the 90s, and just didn't want to be associated with that. So they end up to Bristol Myers. Was involved at Novartis that made it all the way to Gleevec. So Gleevec is a kinase inhibitor. Kinases are central to the regulation of all cells. And so they regulate the growth of the cells, the proliferation of the cells, the division of cells, survival of cells, but there are thousands of different kinases. And so you have to find the right kinase that controls we were targeting, we were targeting PKC, which had nothing to do with what it ended up being, but it was to the issue of serendipity in all of this work. But what we found was a molecule shown up here in the right that inhibits this kinase called ABL. And ABL, when it's mutated and overactive, causes leukemia. A particular form of leukemia called chronic myelogenous leukemia, which 20 was a death sentence. So life expectancy was less than 30%. So 70% of people died within 5 years if they had this. And now they'll survive a normal. It was approved in 2001, and at the time was the fastest FDA drug approval ever for cancer. But it went through a hard road to get there. So chronic myelogenous leukemia only has 5,000 patients per year, which for a drug is a very small sell to 5,000 people product, and it costs $1 billion to develop that product. It's going to be hard to make your money back. So Novartis at the time was not very eager about this forward. Just jump to the punchline. So I actually went to another company to be able to develop these kinase molecules, and was this close to being able to license the Gleevec molecule from Novartis when the first clinical studies results came in, and it was clear it was working fantastically, and everybody who was taking the drug their lives were saved. And so Novartis at that point decided, no, we have to keep this, and it's now a multi-billion-dollar drug. It's so valuable, not just because it works in chronic myelogenous leukemia, but because of some of the side effects, so it also inhibits this other one, c-KIT, which ends up being important for a type of tumor called GIST, gastrointestinal stromal tumors. Also has another 5,000 patients, so it just doubled the market with that. And there are other cancers as well that it's been found to treat. So going from 5,000 patients a year, we're now to over 30,000 patients a year. So this was one of the very first targeted therapies. This and Herceptin were the first two what's called targeted therapies, where you know a specific protein that is mutated in a cancer, and you figure out to inhibit it or activate it, depending on which way you go. And so this was one of the first two of those drugs. And so it did make a lot of press, and for this at this point now a lot of expected to get that from this drug. The third drug that I was involved in is more recent. This is called Padcev, and this is work done that I did at Agencis. And this is an antibody drug conjugate. So an antibody is a protein really structured. And then attached to the antibody is a drug. So these are toxic molecules that are attached through a linker, and the linker is cleavable. And so what happens is the antibody to a receptor on the cell, gets internalized into the cell, and then the cell, the enzymes in the cell cut off this drug. And the drug is toxic, and the way it works is it blocks microtubules that are required for the cell to divide and to separate the chromosomes properly. If you don't do that, the cell, instead of dividing, will actually die. And so it's a way of killing cells that are dividing, which is what you want for cancer cells, because they're rapidly dividing. Targeting something that's on the cancer cells, so it's specific to the cancer. So this is now approved in bladder cancer. It did get accelerated approval, and is able to shrink tumors in 44% of very late stage metastatic cancer that have failed all other. To Nammi Therapeutics, as I mentioned, started this in 2018 with the idea of targeting the immune system to the tumors and blocking the activity of the activation of immune that are common with immune stimulating agents. So to date, we've raised over $17 million from private investors, mostly people in the LA area, and have had $600,000 worth of NIH through SBIR grants. Based in Los Angeles, we actually have our labs at Magnify, which is the incubator at UCLA. It's actually in the CNSI building. If you already. So the overall concept that we are looking to achieve is shown here. The three key features are the redundancy of the pathways of the immune system, where synergistic combinations, so combinations of different drugs that work together to be able to achieve an effect. So that's the first key concept. The second one is that if you really stimulate the immune system strongly, you need to avoid doing that systemically, because it's going to be toxic to the patient. So you're going to have autoimmune diseases that crop up. You're going to have what's called cytokine storms. We know that from COVID these days, where that's why a lot of people die, is from this overactivation of the immune system. And so it's really important to block that activity in circulation. So you want to target it to the tumor. That's the third point here, is a way to target the therapy to the tumor and have it activated only within the tumor environment. So at Nammi, we've developed two different platforms that are able to do this, but in very different ways. So we have the one called Nammizomes and one called masked immunocytokines. So the Nammizomes are nanoparticles, about 100 nanometers in size. This is a schematic of what they look like, but this is an electron microscopy photo of one. And so we have a lead product with this, and a number of other products that are moving forward. This work. These are lipid balls, and what we do is we take a molecule that is able to activate the immune system, attach it to a lipid, and incorporate that into the. As we attach it to the lipid, it stays very stably within that nanoparticle. This into a patient, it'll float around the blood system, not doing anything to the system until it gets to the tumor. When it gets to the tumor, it gets eaten by a tumor cell or a macrophage, and the active is released and is able to activate the other platform that we're using here is what's called a masked immunocytokine. So it's an antibody attached to the ball. The ball at the bottom is a cytokine, interferon alpha in this case, and then a mask that blocks the activity of the interferon alpha. The mask is attached with a cleavable linker to the tumor cell, and the cleaved by enzymes that are on the surface of tumor cells. And so the mask comes off, is activating the immune system to be able to kill the tumor. So that's how these work. So just a brief key concepts of the nanoparticles is this idea of how they target to tumors. So in normal tissues, the endothelial cells lining the blood system, the blood vessels, have these junctions that don't allow large particles to escape. So they flow right through these normal tissues. Tumors are very disarrayed. There are holes, gaps between the endothelial cells that allow these particles to get through. You have lymphatic drainage that would drain those particles away. In the tumor, that prevent them from escaping. So they essentially get trapped there until they're eaten by the tumor cells. Then the immune cytokine, I think more or less pretty effectively the first time, but the idea is that using interferon alpha has already an approved cancer therapy, but it's extremely toxic, so you can't dose it at high enough levels to really have much effect. So our first product is a product that targets a protein on the cell surface of tumors. It's about expressed in about 50% of all tumors. So it's not going to work in everybody, but in a large percentage of patients, it should work. And so it targets the interferon alpha to the tumor. And then the mask will prevent the systemic toxicity of the wild type, the free interferon alpha, so it gets to the tumor, the mask gets, and the interferon is activated. So these two products, the masked immunocytokine and the nanoparticle, are in development now. So we have to do what's called a good lab GLP toxicology study. drug application or IND investigation to the FDA. That allows us to start phase 1. And patients, people who have cancer, late stage cancer in most cases. And our first plan is to test it in multiple myeloma, as well as broadly in solid tumors. And then based on the efficacy from this, we would design a phase 2 study that would expand our patients we look at in those indications and be able to tell us if it's really efficacious compared to whatever does better, then it potentially could get an accelerated approval after. If it looks good, but maybe not overwhelmingly good, then or a larger study where you're looking at larger groups that will be able to give you the statistical significance that you need for approval. So both of these are moving forward. We actually think that there's a good rationale. Singly. So I don't know what the timing is, but that's what I had for my general talk. If there's interest, I have some slides more on cancer and why it's hard to treat, and so that's been made. If there's interest in time. Should I continue, Tom? Or should I take questions at this point?
David, on that last slide, do you think there's eventually will be a cure to cancer, or the project, will it be solved?
So the answer to that is complicated and part of what I have on these other slides, but let me show you this for a moment. Well, let me actually go here. So if you look at what a cancer cell is, what a tumor cell is, and how it gets to be what it is, if you go around the circle, each of these blue statements is something that a cancer cell has developed that gives it special properties over what normal cells have. So if you look at these, they can evade signals from the body that tell cells to not grow. It can avoid the immune system. They are able to replicate indefinitely. Normal cells are not. They're able to promote that causes an environment that is supportive for cancer, et cetera. So they have all these properties that make it perfectly evolved to grow within the body and not to be killed by the defenses of the body. And it also prevents a lot of the therapies from working because they have within them to prevent them from being killed by toxins. Because one of the things that the body will do is build up toxins in areas where these tumors are growing. And so they become resistant to toxins. And that will also lead over to being resistant to toxins that you try to kill it with from outside. We come up with ways to treat each of these different mechanisms that the tumors have. And each of these has some effect. And so we chipped away, and each of these will save lives, but it's, a few patients here, or perhaps it just does, saves 6 months on average for a broad percentage of the population. And so to get longer, then we start looking at combinations of these. But when you start combining these, then you start getting toxicity. So that's a long answer to your question, but the is a long answer because it's a very complex question, much more complex. Understanding more and more of the complexity. But that said, I do think that the closest thing we come to a general cure is really going after the immune system. So in the past 5 years, there's been tremendous advances within the area of immunology, cancer immunology in particular, to stimulate the immune system to kill tumors. And it's been very effective. Though it's still only effective in about 20% of cancer patients across all indications. Some indications like melanoma are very reactive. So it's more like 60% of melanoma patients who would have died before are now being saved from immunotherapy. And that's really where Nammi Therapeutics comes in, is we're trying to create the conditions with the environment that will allow them to be sensitive to immunotherapies. So being able to percent to 50%, 60% or more. So yeah, we're not going to get to a cure anytime soon, but we are chipping away and making it, if not cured, at least something that you live with rather than. Other questions? Here's some of the progress. So if you look at overall incidence of cancer, so the orange is the average, pink for female, blue for male. The incidence to about somewhere in, and then has started coming down. It's definitely come down from lower. So this is when the war on cancer was initially started, was in around 75, and you can see the mortality rates are definitely trending down, and we hope.
Dave, is this cocktail you worked on, is this the Magic Johnson cocktail?
Yes. Yeah.
And then do you see a prognosis for pancreatic cancer being eradicated in our lifetime?
So there are two main issues with pancreatic cancer. One is that currently it's diagnosed way too late. So by the time you know you have it, it's typically a death sentence, because there aren't good symptoms that people can recognize as being a real problem until it's too late. So there's a lot going on to try to identify it earlier. And I think we'll be able to do that for a large population. And then also through some of the therapies, our Quixel 138 AM works very well in pancreatic cancer preclinically. So but I do think that early diagnosis is going to be a key in shorter term. Yeah.
Thank you.
Yeah.
I've got a question. I believe I remember back in the early 70s, I was told that people who are over 65, half of them died due to cancer. I don't know if that was completely erroneous or what, but with these reductions here, what do you have any idea how many people over 65 died due to cancer now?
So it is still one of the top 3 killers of all people. 65 even more so, because that's where most people that get cancer are going to get it. It's over 65. So it's probably a little bit better than 50% now, but people are living longer as well. So it's an improvement. And it's also important to distinguish between dying with cancer and dying from cancer. So especially prostate cancer, you can look at, by the time you're 90, you're certainly going to have prostate cancer. Most people will die with it, not from it.
Mhm. That's a good answer, yeah. David,
Sure. Yeah.
your financing to date has been done with individuals. Is that it basically?
You have to
And
have institutional VCs on board.
Did I see 17 million is what you've raised to date? Is that right? About?
Yeah.
Are you getting to the stage or where you can go? Because you're going to need more money before long, aren't you? Going to the venture capital route?
Yes, that's what I'm involved in now is reaching out to venture capital to bring that in. That have had similar approaches as us. They've gone from a Series B, which is what we're looking at, to an IPO in about 6 months. So we're sort of hoping to be on that timeline.
Good. Was another question, was Gilead part of that HIV cocktail?
They have one part of it, not the protease inhibitor though. Yeah. They have the RNA.
Dave, Mark Rogo. Obviously, all the testing, the clinical trials and everything involve masses of people and tons of labs and lab work and personnel and technicians, but you got 6 employees. How are you doing all of that?
We're not yet in the clinic, so we have the people we need to do the testing in vitro, so in test tubes. For animal studies, we are outsourcing that. For manufacturing, we're outsourcing. So we have people in-house that can manage the outsourced work. So even though we're only 6 people, we're doing work of probably more like 50 to 60 people, based on how much we're outsourcing. Yeah.
You're going to have time for Rotary meetings every Thursday?
Well, that's one of the beauties of outsourcing, right? Somebody else is doing the details. So
Okay. Thank you. Very impressive. I really enjoyed this.
thank you.
It's Dick Thompson. Any observations on prevention on a broad scale?
So preventing getting cancer to begin with, right?
What are most of us doing that would make us more liable?
I mean, the big things, right, that have been in the works are, don't smoke. That's a clear one, and protection, those kinds of things. But there's always going to be some, even without those. People are born with tumors. There's just an ongoing mutation that occurs even in the absence of these toxins, which do accelerate it. But there's always going to be a baseline that happens. What you do with your lifestyle.
Thank you, Dave. You're a hero to a large group of people now.
Thank you.
Are there any other questions for David?
Yeah, I had the male incidence and male mortality is that because males have a tendency to dissipate more and also work in fields that subject themselves to chemicals and fumes and all of that?
I think that's a large part of it, especially in the earlier years here in 75 to 95. I think the balance between those is changing, and maybe you can see that toward the end of the curve here, where exposure from males and females in the workforce is becoming more even, and so you can see that reflected in the incidence. Yeah, it's hard to say at this point whether there's an intrinsic difference. There's some thought that there may be some level of intrinsic difference. Interesting that the tumors are able to avoid immune clearance. So the body's immune clearance of tumor cells is using mechanisms that are used by a fetus growing in a pregnant woman. So in order for a fetus not to be attacked by the mother's immune system, it has to have all these defenses that prevent it from. And the tumor takes advantage of those mechanisms to escape the immune system as well. So whether or not, the differences between of what a woman has to deal with in pregnancy could affect to fight cancer, maybe different.
Well, David, these very interesting subjects, probably far over my head about some of the technical things, but it's interesting to see the progress your companies have made. Let me ask you one final question, if we could. We're ready to end. How much confidence do you have in the vaccinations for COVID-19?
Confidence in terms of whether they're working. I have a lot of confidence. I think it's
Yeah.
The tests have been done and shown that they're quite effective at treating or preventing serious illness and death. In terms of getting some level of infection that might be visible to some extent, that's a little less clear how effective they are.
Yes.
But in terms of preventing death and serious illness, it's clear that they're really.
So obviously you've been vaccinated.
Yeah.
Yeah.
Yeah, and I'll go get my booster pretty soon as well.
So thank you very much for this presentation and Craft Talk. We appreciate it very much. And in your honor, we're going to be making a contribution to the Westwood Village Public Library at the end of the year. So thank you very much. Next week, we have Professor Emeritus Elizabeth Breen from UCLA. She'll be speaking to us. So be sure to tune in. And with that, we'll call a meeting to an end.